So chapter 10 is about muscle tissue, and this is related to physiology and also some histology of the muscle. As you remember, we have three different types of muscles: a skeletal muscle, cardiac muscle, and a smooth muscle. Of those three, only the skeletal muscle has voluntary movements. The other two has involuntary movements. So the function of the skeletal muscle, there is some common properties of the skeletal muscle. One is excitability, which is also called responsiveness or irritability. And that is because the skeletal muscle has change in the membrane potential. So it has the ability to change its membrane potential. And that is due to its response from nervous tissue. That excitability will cause contractility, and the contractility will produce tension. And that is the main function of the muscle. It's also extensibility, which means it can stretch out, and elasticity, which it can bring it back to the normal length or even shorter length. So spring back to the original length. There are different functions of the skeletal muscle. One is producing movement, which is the main function. So, for example, walking. It can be writing, moving your head, so some things like that. It's maintaining for posture and body position. Examples are including a contraction of the neck muscles. That's how your head is up when you are awaking. When you are awake. The contraction of back muscles, and this is how it keeps your posture. The contraction of leg and thigh muscles. This is how you are standing. So different body postures. It supports soft tissue, protects some soft tissue. It's also located in different entrances and exits. For example, skeletal muscle is located around the eyes and around the mouth, so you can open or close your mouth. It's also located around the anal and genital area or urinary area, and that is a way it's protecting those areas from entry of any insects or foreign body into the body, and also it's important for urination and defecation. It maintains the body temperature, so during muscular activity there's a lot of ATP is produced, and that will help increasing the body temperature. If it is really cold, you start shivering, and the shivering is actually contraction of the skeletal muscle, and that way the body temperature increases. It stores some nutrients. There are calcium and iron is there, but most importantly, lots of proteins are there, and proteins can be broken down to amino acids. And when the body is starving and not getting the energy through the lipids and carbohydrates, the amino acid can be broken down and can be used as a source of energy. This is the basic organization of the skeletal muscle. So if this is a skeletal muscle, or this one, any of these ones are skeletal muscles, which looks like this, it is covered by a type of connective tissue, and that is called epimesia. The skeletal muscle is made of these bundles, and these ones, these are called muscle fascicles, and the muscle fascicle is covered by another type of tissue, and that is called perimesia. That is a connective tissue again. Then the muscle fascicle is made of multiple muscle fibers. So these are the muscle fibers. That they are covered by another type of connective tissue, and that's called endomysium. So this is the muscle fiber, and it is wrapped by or enclosed by endomysium. And this is how you see a muscle fiber, and this is the endomysium that is there. Endomysium also covers the myosatellite cells that you can see here. Inside the muscle fiber, you see a lot of these other fibers, and these are called myofibrils. There's a little bit more information about the organization of the skeletal muscle. So epimysium that wraps around the whole muscle. It contains its own blood supply and nerve. So you can see the blood vessels are here, and this is a nerve. And it is made of collagen fibers. Then, as I said before, the muscle itself is made of multiple muscle fascicles. These are the muscle fascicles. So this is one muscle fascicle, and that is wrapped by perimysium, and perimysium contains, again, smaller arteries and nerve fibers, and is made of elastic and collagen fibers. Then the muscle fascicle is made of these muscle fibers, and each muscle fiber, which is here, is now wrapped by different type of connective tissue. This elastic connective tissue, and this one contains capillaries and myosatellite cells. Inside the muscle fiber, which is the same as the muscle cell, you have myofibrils. So this is the myofibrils, and this is a larger view of a myofibril, and myofibril is wrapped by sarcoplasmic reticulum. This thing that you see, this blue area, this is sarcoplasmic reticulum. And the myofibril is made of a thick and thin filament. So this blue line that you see, this is the thick filament, and the red one that you see, this is a thin filament. And from this area to this area, it's called sarcomere. So one muscle fiber has a lot of myofibrils, and one myofibril has a lot of sarcomeres. So the muscle fibers develop from mesodermal cells. These are embryonic cells, and they convert and they become myoblasts. So these are the myoblasts. Many myoblasts fuse together, and they become an immature muscle fiber. But sometimes some of those myoblasts will not fuse, and they stay outside near the immature muscle fiber, and they are called myosatellite cells. These are the stem cells. So later on, these many myoblasts that fuse together, that becomes a mature muscle fiber, which is really long. A nerve cell is the longest cell in the body, and the muscle fibers are the second longest cells in the body. So they contains hundreds of nuclei. Again, it varies from one muscle fiber to another, so some may have less than 100. And it is striated. So these lines that you see, this is the striation, and these are the striation. These lines that you see, again, you see that under microscope striation. That is because of overlap of thin and thick filaments. And these dots that you see, these are the nuclei. So it is multinucleated, striated, and it is voluntary movement. One more thing is about the myosatellite cells. So in case the muscle is injured, these myosatellite cells become enlarged and then divide, and they fuse with muscle fiber. So it helps repairing the muscle fiber. If the muscle fiber is totally damaged, it cannot be repaired. So this is unlike epithelial tissue or connective tissue that can be repaired, but muscle cells and the nerve cells, if this damage is severe, they cannot be repaired. There are some exceptions in the nerves, but muscle cells and nerve cells, usually then they don't repair once they're damaged. So there are specific characteristics of the skeletal muscle. One is called sarcolemma. Sarcolemma is the plasma membrane, the plasma membrane covering the cytoplasm, and the cytoplasm is called sarcoplasm. And one of the features of the muscle fibers is that it has a difference in the membrane potential. So inside and outside of the cell has a different membrane potential. Once it is stimulated by a nerve, it can transmit the electrical activity inside the muscle fiber, and that will cause contraction of the muscle fiber at the same time. So the whole muscle fiber will contract. It's not just random parts of it, the whole muscle fiber will contract. There is also the T-tubule that is there. So the T-tubules, these are the T-tubules that you can see. This is the invagination of the sarcolemma or plasma membrane. So this is the plasma membrane, and this is invagination. It's like a glove that goes inside the cell and wraps around these myofibrils. So these are the myofibrils. So wraps around the myofibrils. It goes all the way everywhere. And these are all also the T-tubules. The function of the T-tubule is to transmit electrical activity inside the cell. And once inside the cell, all of the muscle fibers are activated, all of them can contract at the same time. It's not just stimulating the peripheral myofibrils, it will stimulate all myofibrils of a muscle fiber at the same time. So this is why it goes all the way. If these T-tubules were not there, then only the superficial, these ones, would have been activated because they are close to sarcolemma, but the deeper ones would not have been activated. So that's why you have this mechanism. So the sarcoplasmic reticulum, this thing that you see, this blue structure that you see, this is the sarcoplasmic reticulum. This is the same as endoplasmic reticulum. It contains calcium, so it is an important source during muscle contraction. Without the calcium, the muscle cannot contract. So the calcium that is in the sarcoplasmic reticulum is released, and then it will contract. As you can see, these areas, the end, it become enlarged. So these are the enlarged chambers. These are called cisterna. Cisternae is the plural form. So each sarcoplasmic reticulum has, an expanded area, this is the T-tubule in the middle, and two expanded area, two cisternae together. All three of them are together. That is called the triad. So triad is the T-tubule and the two cisternae of sarcoplasmic reticulum, which is unique to the skeletal muscle, because the cardiac muscle and smooth muscle, they don't have the triad. Now is the myofibril itself. So you can see this is the inside of the muscle fiber. So it contains a lot of these myofibrils that is wrapped by the sarcoplasmic reticulum and by the T-tubules. So this myofibril is made of the thin filament, which is this red one, and the thick filament, which is these dark blue ones. And this is important for the contraction of the muscle. So what is a sarcomere? Sarcomere is from one Z disc, this is our Z line. This is from one Z line or Z disc to another one. So this is a sarcomere. So this is a repeated functional unit of myofibril, which in turn is muscle fiber also. So it's a functional unit of the muscle fiber. And one muscle fiber, as I said before, contains a lot of myofibrils, and one myofibril contains a lot of sarcomeres. So in a sarcomere that you see, there is a few different structures are there. This thing that you see in the middle, this is called M line. This is M line, okay, and this is the M line is connecting the thick filaments together. So this is the thick filaments that you see, this dark red area, and this area that contains only the thick filament, this is called the H zone, contains only thick filament. Then this area that contains a thin filament and thick filament together, this area, this is called the zone of overlap. Zone of overlap. So what is the A band? A band is the zone of overlap, so one on each side, and in the middle you have the H zone. So from here to here, that is the A band. And finally, the I band is an area that contains only the thin filament. So in the middle of the I band, you can see the Z disc, but there is no thick filament in this area. So what is the thin filament and thick filament? So look at this image again. So you can see a sarcomere here from one Z line to another Z line. In the middle, this image actually is nicer, you can see it easier. You have the M line that connecting the thick filaments. The thick filaments are connected here. The area that only contains thick filament is called the H band. And then this area from here to here that contains both thick and thin filament, that is called the zone of overlap. And from here to here, then is the A band. Okay, and I band is this area that does not contain any thick filament. So now if you look at it closer, let's go to the thin filament. And the thin filament has 4 different proteins are there. One is the F-actin. So F-actin is a strand, it's a twisted strand that are made of G-acting molecules. So there's a lot of G-acting molecules that are twisted, and that in the middle it is connected by this protein, that is the nebulin, that is connecting the G-acting molecules. The G-acting molecules contains an active site to this yellow area that you see. This is the active site of G-acting molecules, and that is covered by this protein, and that is called tropomyosin. So this one, that is covering the active site of myosin. So normally at rest, the active site of actin, I mean. So normally at rest, when the tropomyosin is covering the active site of actin, then the myosin cannot come and attach to the actin. So that's one of the functions of tropomyosin. And one tropomyosin can cover 7 active site of actin. Then is another protein here that is called troponin. The troponin, one part of it is attached to the G-acting molecule, another part is attached to the tropomyosin, and there is also a free area here that can attach to calcium. So normally when there's no calcium, nothing is happening and the muscle is totally relaxed. You also have something else that's called actinin, and actinin is a protein that is connecting two thin filaments. So one from each sarcomere is connecting the two to each other. And also there is another protein that is called titin. Titin is connecting the thick filament to the Z line. Thick filament to Z line. Okay, so you see the thick filament here, and it's connecting it to the Z line. So the thick filament is made of multiple myosin proteins. So the myosin protein contains a tail. The end of the tail goes to the M line, and then it connects with the tail of the opposite side myosin. It has a hinge, and this area is moving the head, and the head has there's 2 heads here. In a smooth muscle, there's a lot of heads, and the skeletal muscle, there are 2 heads. And these heads are toward the thin filament. There is also the titin that I told you before, and this is one of the very big proteins, one of the longest proteins. It gives elasticity to the myofibril, so it is coming all the way from the M line, goes inside, and then it's here is twisted, and that goes and connects with the Z line. So in case if the muscle is stretched, then the titin can contract, or if the muscle is contracted too much, then it can spring back. So the main function of titin is to bring back the muscle to the normal length. Sliding filament theory is that I can give you two different status, and then it's easier to understand. So normally when the muscle is relaxed, you can see the H band is wide, and also the I band is wide here. There is also zone of overlaps are there, so the thick and thin filament can be close to each other. And when the muscle is contracting, what happens that these heads of the thick filament can attach to the thin filament, and the thin filament moves toward the M line, toward the middle, and in that case the H band width decrease. Okay, also the Z line, the I band decrease here, but the zone of the A band size remains the same, and the zone of overlap size or width increase. Okay, so the I band decrease, the H band decrease, the I band decrease, the zone of overlap increase, but the A band will remain the same. So when the thick filament attaches to the thin filament is moving toward the midline. That is the sliding idea. So that's where the muscle contracts, and during relaxation, then it goes back to the length that it was before. So a different topic that's called neuromuscular junction. Neuromuscular junction means a connection between a neuron and a muscle. So if you look at it here, you see that this is the end of a neuron, and this is the plasma membrane of the neuron. This is called the presynaptic membrane. Okay, and that area contains also these vesicles. The vesicles contains acetylcholine. This is acetylcholine. Okay, contains acetylcholine, which is a neurotransmitter. It's a chemical. Then you have this is a plasma membrane of the muscle. This is the T-tubule of the plasma membrane. This is called also postsynaptic membrane. And the area in between this area, this is called synaptic cleft. Synaptic cleft, that is a space between presynaptic and postsynaptic membrane. You also have these receptors that are responsive to the acetylcholine. So I will show you what will happen to those receptors. And you have some enzymes here. These are called acetylcholine esterase that breaks down acetylcholine. So the motor end plate is the area that the nerve connects to muscle fiber, and that is the area that the muscle fiber can be activated. So what happens here, an action potential arrives. An action potential, in case if you forgot from Bio 1, is that changing in the membrane potential of the neuron, and that will cause movement of sodium inside the cell. Inside the cell become more positive. So action potential comes here once it gets to the presynaptic membrane. Then calcium enters the cell, which is not shown here, and that calcium will cause fusion of these vesicles into the presynaptic membrane. Once they fuse here, then the neurotransmitters, which are acetylcholine, they are released in the synaptic cleft here. Now they go on attached in these receptors, and these are the sodium channels. So once they are attached to these ones, the sodium channels open up and the sodium enters the cell. Once the sodium enters the cell of the skeletal muscle fiber, inside the cell become positive, and that is a change in the membrane potential. So that will cause, initially graded potential and then an action potential. So here you will have the T tubules are also activated here. Then you have these acetylcholines are attached to acetylcholine esterase, and that acetylcholine esterase breaks it down, break down the acetylcholine, and it will be recycled back. So when the next time there's a need for it, then it can be used right away. So excitation-contraction coupling, what does it mean? It means that the nerve has to be excited or stimulated, and that stimulation comes to the muscle and cause contraction of the muscle. So these two has to happen back to back, and if that does not happen, then no muscle activity will happen. So here is the neural activity. This is a neuron, and this is the branches of a muscle fiber that is attached to the sarcolemma of the muscle fiber. So then this the ending, they have these vesicles, and this is the end of this neuron. So this is called axon, and they have these vesicles there. This is contain acetylcholine. So when there is a stimulation there, the acetylcholine is released, and then activating the plasma membrane, and that will go to the T tubule. So now the T tubule is located close to the sarcoplasmic reticulum, and if you remember, the sarcoplasmic reticulum is covering or surrounding the myofibrils. So here the sarcoplasmic reticulum contains calcium, and that calcium is released, and that will cause muscle contraction. So up to here, when the neurotransmitters are released from an upcoming action potential of the nerve, and then the plasma membrane of the skeletal muscle is activated. That's the excitation part, and then once the calcium is released, and that will cause movement of the thin filament over the thick filament, that is the contraction part. So if this excitation is not there, this contraction will not take place. Okay, so what we see here is that you can see the F strand, F actin strand, and these are the G acting molecules that contains the active site. Normally the active site is covered by tropomyosin, and you also have troponin here. So there is no calcium there, and the active site is covered by tropomyosin, so there is nothing happening. No connection between myosin and actin will happen here. Once the calcium enters from sarcoplasmic reticulum, as we saw before, so now the calcium attaches to troponin, and troponin moves tropomyosin. So once the tropomyosin is moved, then what will happen here? The active site of actin is exposed. You can also see here a nebulin that are connecting these G-acting molecules. Then what will happen that the head of the myosin comes and attaches to active site of actin, and this action is called cross bridge. So cross bridges when the head of the myosin comes and attaches to the active site of actin. If you look at it from sideways, so you want to know what's going on in the contraction cycle, here you have the F-acting strand, and these are G-acting molecules. This is the nebulin, and this is the tropomyosin that covers the active site of actin, and there is a tropomyosin that is keeping the troponin that's keeping the tropomyosin there. So when there is no calcium, nothing is going on. Okay. Also, you see that you have the myosin, and you have the head of the myosin that contains an energy molecule, which is ADP plus phosphate. So once the calcium enters the area, what will happen that the calcium again attaches to the troponin. Troponin moves tropomyosin, and now the active side of actin is exposed. The active side of actin is exposed here. Once the active side of actin is exposed and the myosin head also has energy that goes and attaches here. So that will cause the cross bridge. Okay, so that attachment of the head of the myosin to the active side of actin. Then the head of the myosin moves the actin toward the midline. Okay, it moves it toward the head midline, and that will cause a release of ADP and phosphate. So now this is the sliding filament theory that I talked about it before. This is what's happening. So the H band is decreasing, and the thin filament is sliding over the thick filament. Then what happens that ATP, another ATP molecule comes here, attaches to the myosin head. So this attachment will cause detachment of the myosin head from the active side of actin. Okay, detachment. Now the ATP hydrolyzes again to ADP and phosphate. That will result in the movement of the myosin head back to the same way it was. So when it goes back to that and the calcium is still there, it means the active side of actin is still open. Then the myosin head goes back and attaches to the active site of actin again and moves actin again toward the midline. So this happens many times. So when it moves it toward the midline, that is called power stroke, moving it toward the midline. What will happen during the relaxation of the muscle? So that depends on the duration of the neural stimulus. So if this, because skeletal muscles are under your conscious control, as long as you want to do an activity that is a neural stimulation is there. But there should be other factors involved as well. So there is the amount of the calcium there. For example, not enough calcium, the muscle contraction may not take place. And the amount of the ATP is important, because even if you want to do something, but not enough energy there, so the muscle cannot contract. For example, you want to walk 5 miles and then you are getting tired and then you cannot go all the way. So the amount of energy is not there to go all the way, despite the fact that you want to do it. So once the neural stimulus stops, okay, then the calcium goes back to the sarcoplasmic reticulum, so the calcium level falls, and then it means that the troponin is not attached to the calcium level anymore. So now here is the troponin goes back to its normal site, and the tropomyosin goes and covers the active side of actin. So the myosin head cannot attach to the active side of actin anymore. And this is this whole summary of what I talked about it, so I will not go over it again. So on this side, this is the contraction that is happening, and this is the reverse of the muscle relaxation. So you can stop the video and you can see what is going on and try to explain what is going on. So this, I purposely, I don't want to explain it, so you can explain and see how much you learned so far. So a different topic that we want to talk about, this is about the muscle tension. The muscle tension depends on the resting length of the muscle fiber. Okay, so at the time of stimulation, how much is the length of the muscle fiber? And the second thing is about the frequency of the stimulation. So let's talk about the frequency of the stimulation. First, there's 5 different ways the muscles are stimulated, depends how fast the signals are coming. The first one is called twitches. Twitches are happening only once. These are spontaneous, involuntary. You did not decide to do it. It can happen locally at the lower motor neuron. So lower motor neuron means a spinal nerve or a cranial nerve. So it's outside of the central nervous system, outside of the spinal cord and the brain. And one motor unit will contract. So I will come back to the motor unit, which is a few muscle fibers. They contract and then they relax. So depends on the type of the muscle. So this is one time is contraction or relaxation. These are the muscles in the lower limb. So the duration of that depends how many muscle fibers are activated. So twitch is what? One time, involuntary, local stimulation of the muscle, and that's it. Trip is that when the stimulus is less than 50 per second. So what happens here, there's a stimulus happens, it can cause increase in tension, then the muscle goes back to relaxation, then it increases and it goes back to relaxation. But every time the stimulus is there is some increase in the amount of tension, because not all calciums can enter back to sarcoplasmic reticulum, so there is some increase in the tension. But you can see the tension is very low here. Wave summation is the same idea, but the amount of stimulation is more than 50 per second. So here the stimulation is there, so the tension increase. The muscle tries to go back to relaxation, but it cannot go back to the baseline. Here it went back to baseline, but here it cannot go back to baseline again. So then you have another stimulation, so the tension increase until you stop. Then it goes back to the baseline and relaxes. In the incomplete tetanus, same idea as summation, but you continue stimulation until you reach a plateau phase. And here the muscle tension is much higher than before, but the muscle still relaxes a little bit. In between the stimuli, the muscle still relaxes a little bit. And in complete tetanus, it's very, very, very fast. The stimulation is so fast, the muscle cannot relax. So the tension increases very rapidly, and it goes all the way to the maximum, and it stays in the maximum until you go back to relaxation. So this happens in the muscle physiology. It can also happen during different type of muscle activity, and also it can happen in abnormal states like tetanus, which is a bacterial infection. So the second factor was the length-tension relationship. So if you look at it here, this area, that area produces 100% maximum tension, and this area is called optimal resting length. So what does it mean here? That for all the heads of the myosin, you have an area of the actin, that the head of the myosin can attach to the actin and it can move the thin filament toward the midline. Okay, all areas. But if you look at it this way, so now the thin filaments are all the way to the midline there. The head of the myosin can attach, all heads of myosin can attach to the thin filament, but there is not much H zone left here, no H zone. Okay, so it can produce some tension, but not as much. Look at it here. The thin filaments are across the midline. There is no H zone there, so it cannot produce any tension here. For example, your muscle is totally contracted. It cannot produce more. Okay. The other way around is that here in this area, that some myosin heads can attach to the thin filament. Some of them. See, these ones cannot do it, but these ones can. So now, when the muscle contraction takes place, that these thin filaments can move this way toward the midline, but only a small part of it. Then you need another one to move it a little bit more. So this is why the amount of tension is not as much. And if the muscle is very stretched, then there is no connection between the thick filament and thin filament. So even if you stimulate the muscle, these thick filament the heads, the head of the myosins cannot attach to the thin filament. It cannot attach to the actin. So nothing will happen. So that's why the tension is almost zero here, or it will go to zero if the muscle is stretched too much. What is a motor unit? It means that one nerve fiber can innervate multiple muscle fibers. And this is good for efficiency, because if you have one nerve fiber for each one muscle fiber, first of all, the thickness of the nerve will be very big, the same size as the muscle. And the second thing, you need a lot more coordination. And sometimes you don't need coordination. A lot of muscle movements are basic, so you don't need a lot of coordination. So once the signal is sent, multiple muscle fibers can contract at the same time. So this is why the whole thing is called motor unit. So one nerve fiber plus multiple muscle fibers that responding to the nerve stimulation. And recruitment is something different. So when you're doing some kind of activity, when you're moving something, when you want to overcome a load or a weight, initially small fibers are activated. Okay, that produces a little bit tension, but if it cannot overcome the load or the weight, then you have more fibers that are involved. If it can overcome, that's good. If it cannot overcome, then the bigger fibers are involved. Finally, it may overcome the load, but this produces a lot of tension. And as you can see, the time, it gradually increases. All of them will not happen at the same time. It gradually increases. And that gradual is important for a smoothness of the motor activity, and also to preserve extra energy. For every movement, you do not need to activate all muscle fibers. You only activate more muscle fibers if you need it. If you don't need it, you don't need to do it. For example, if there is a crime somewhere, you don't need to call all the cops of the whole county in that area. You don't need it. You may need 1 or 2 or 3 cops there. The same idea here. So once then there is a need for it, then you can recruit more muscle fibers. Classification of the muscle contraction. One is called isotonic, and the other one is called isometric. So isotonic means the tone is the same, but the length of the muscle is different. So there's 2 types here. One is called concentric, and the other one is called eccentric. For the isotonic contraction, for example, lifting something or walking and things like that, these are examples of isotonic contraction. So in the concentric one, the amount of tension that is produced in the muscle can overcome the load or the weight, and the length of the muscle decrease. Okay, for example, you can lift something, and this is not heavy, and you can lift something and then you can carry it. The other way around is that the load is really heavy. You cannot lift it. It's very heavy. And in that kind of a case, the muscle can still contract, isotonic, but because of the gravity, the length of the muscle increase, because the amount of tension that is produced in the muscle, it cannot overcome the load. Okay. For example, you want to go to gym and you want to lift something. If you're lifting something small, it's easy, you can do it. But if you're trying to impress your friends and try to lift something heavy, and then you know that nobody will be impressed with your movements in the gym. So even if you try to do it, nothing will happen. So you will have an eccentric contraction. Compared with me, if I go there, I will have concentric contractions, right? I'm joking. So the other one is isometric contraction. So the isometric contraction, the length of the muscle remains the same. So this is important for posture or keeping a specific body positions. So here is that this is important for resistance training. So the amount of the tension that is produced is different, but the length of the muscle remains the same. The load and the speed of contraction, they are inversely related. So if it's a small load or weight, it takes less time for the muscle to contract and increase tension. So once the tension is increased, the length of the muscle decrease, and you can keep the tension for a longer period of time. But if it is a bigger load, it will take longer time to start, and you cannot keep it for a long period of time. So the amount of tension that also produced may be not as much, because the load is so high that even if you try to produce a lot of tension, the muscle cannot contract as much, so that the length of the muscle may not shorten enough. That again depends on the situation and the type of activity, but here we are talking about the timing and the speed of the contraction. So here it will be shorter, and it takes longer time to start. Okay, so there's a few different definitions here about the motor unit and the tension production. One is called maximum tension. So in the maximum tension, all motor units contract at the same time. They reach maximum tension, so the highest tension that is possible. The problem with this is that you cannot continue having this status, because all the energy that is in the muscle fibers that is used, so it's only working for a very short period of time. The amount of tension will be high, the amount of force will be high, but the duration will be very, very short. Compared with the sustained tension, here you do not use all your muscle fibers. You use some of them, and that will increase tension, but it will not be maximum tension because you didn't use all muscle fibers in the whole muscle. Once those muscle fibers are tired, then the muscle fibers that were relaxing will take over, and those muscles that were tired will get their oxygen, and then they will have their ATP production, and they were ready to take over again. So then you can continue doing this for a much longer period of time. And the muscle tone is that very, very few muscle fibers are active. So this is for body positions. For example, you're sitting and your head is straight, or when you're standing, you're not falling down because the back muscles and the lower limb muscles are contracting, but not all of them are contracting because you get tired very fast. So some muscle fibers are contracting, and that keeps your posture and body position. So this is without movement. It's just for a specific body position. What will happen when the muscle is relaxed? When the muscle activity stops, the return of the muscle back to its normal length depends on a few different factors. One is the elastic forces that are there. So you have ligaments and tendons that has some elastic fibers, and that will bring the muscle back. You also have the titin protein inside the myofibrils that is helping there. The other one is the opposite muscles. So for example, you're abducting, and then you have other muscles for adduction. So those muscles will help bring it back to the original position. Or for example, you're flexing and you have the extensor muscles that are opposing those movements. And the last one is the gravity that's always helping. For example, you're lifting your hand or your limb for a while, but you cannot do it forever, and the gravity brings it back to the original position. So it's a different topic. It's about the energy for contraction of the muscles. The muscles use a lot of energy, so it needs a big source of energy. Normally when it's at rest, it is storing energy, and when during the activity it breaks down the ATP, but also produces new ATPs, depends on the type of the muscle fibers. So when it is the person is resting and not doing any kind of activity, the ATP that is produced combines with creatine, and that will produce ADP and creatine phosphate, which is a molecule as a source of energy storage. But during the activity, what happens that the ATP that the muscle is producing, instead of being stored as a creatine phosphate, that converts to ADP and phosphate. So that hydrolyzes to inorganic phosphate and ADP. The ADP can also combine with creatine phosphate, which was stored as a source of energy, that brings it ATP. So then ATP can be used again as a source of energy, and then this will produce creatine, which in the future you may have a phosphate attached to it, so this can be used for future as a energy storage, but for now you need ATP. So this will break down the creatine phosphate to produce ATP, and the enzyme that is helpful for that is called creatine kinase. If the activity continues, then you need to have a glycogen that breaks down to glucose, and also fatty acid that should be broken down, which I will talk about it in a second. Okay. So what happens there at rest? Okay. This is the blood vessel, and this is the muscle fiber. So there's oxygen is coming, and as you know, mitochondria needs oxygen for its normal activity. There is also glucose coming, and the fatty acid is coming. So fatty acid comes inside the cell. The mitochondria has a lot of oxygen, and the fatty acid can produce a lot of ATP. You don't need glucose to be broken down. So now this glucose goes and stores as glycogen, and for that you may need some ATP, and so you need that ATP coming from the mitochondria to convert glucose to glycogen. So glycogen is stored in the muscle cell, and when you need it, then it can be used as a source of energy. Also, there is a creatine is there, and creatine combines with ATP, and the phosphate goes to creatine, and that becomes creatine phosphate, and then it's another molecule for energy. So you have two molecules for energy as a storage, and you can use fatty acid at rest. So if you do some moderate activity, and the moderate activity oxygen is still there, what happens is that the fatty acids are coming, broken down in the mitochondria, and that produces carbon dioxide, so that will go back to the blood, and that will produce energy. But also you may need glycogen. So glycogen can be broken down to glucose, and glucose in the cytosol and the cytoplasm broken down to pyruvate, and that will produce 2 ATPs, not much, just 2 ATPs. And pyruvate can enter mitochondria, and now it can produce 30 ATPs. So there's a lot of ATPs that can produce. But one fatty acid, minimum, it can produce 129 ATPs. Depends on the length of the fatty acid. This is the minimum it can produce. So the fatty acids can produce a lot more energy than glucose, but still it's a good source of energy. Then what will happen that if you're doing a lot of activity, not enough oxygen is going there. So now mitochondria may not have much function, and that's a problem. So here is the glycogen broken down to glucose, and then in the cytoplasm you produce only 2 ATPs. So from glucose to pyruvate, only 2 ATPs. Also, you can break down creatine phosphate, and you can produce some ATP there. But here the pyruvate will not enter mitochondria because it does not have enough oxygen, and that will convert to lactate, and that makes everything acidic because of production of the hydrogen. and that lactate goes to the blood, so that will decrease blood pH. So that during the recovery period, the muscle has to relax, and then the oxygen comes, and when the oxygen goes into mitochondria, then the mitochondria can produce activity. So what happens to the lactate? Then the lactate goes to the blood, from there it goes to the liver, and through the gluconeogenesis, it produces new glucose, then the glucose can go to can store in the liver, or it can go to the muscle and then it stores as glycogen. So glycogen storage happens in the liver or in the muscle, or if the body needs glucose, it can broken down. For example, it goes to the brain. So muscle performance depends on the force. Force is the amount of the maximum amount of tension that is produced. It means that all muscle fibers contract at the same time. For example, you're lifting something very heavy, you're lifting some weight, for example. Endurance is that how long you can do an activity. This does not produce a maximum amount of tension. So some fibers are active, some fibers are not active. For example, you're walking, you're jogging, you're swimming. And these two, the muscle force and endurance, or muscle performance in general, depends on the muscle fibers and also physical conditioning. Physical conditioning is related to the physical activities that you do, or the kind of athletic activities that you're doing. There are three different types of muscle fibers. One is fast muscle fibers. They contain a lot of glycogen, and that glycogen can be broken down, and that produce a lot of energy, and this is why these muscles can contract very fast. But because they do not have a lot of mitochondria, then they cannot continue contracting. So it can produce maximum tension, but it cannot use sustained tension that much, because it will be tired very fast. The slow fibers, or the other way around, does not have enough glycogen, but it contains a lot of myoglobin, which has oxygen, and also it has more blood supply, and that has more oxygen for mitochondria. So it can continuously produce ATP. So this type of fibers cannot produce maximum tension, but is good for the tone and for sustained tension. And intermediate fibers are just in between. For humans, the majority of human muscles are pink ones. It's a mix of them. Some muscles, for example, the hand muscles and the eye muscles, they cannot be slow fibers, because it requires a lot of activity very fast. And some muscles, like back muscles and leg muscles, if they are used for a tone, then they can be used as a slow fiber. So it depends on the type of activity and the location of the body. These muscles are a little bit different than the type of fibers. The type of fibers also related to underlying genetics. So that's why athletes are going through the same type of training, but some are better than the others because they may have better genes. Okay, muscle hypertrophy. So this is one of my pictures, as you can see it. If you are doing a lot of training, the training can be due to like weight training, or it can be other physical activities from work or anything else. Then the number of the myofibrils increase. Also, the glycogen and mitochondria in the muscle increase. But the amount of, or the number of the muscle fibers remain the same. The diameter of the muscle fibers increase, but the number of muscle fibers remain the same. If you are not doing enough physical activities, then that will happen to muscle atrophy. So here the size and the number of the myofibrils decrease, and also the amount of muscle fibers decrease. And that can be from lack of activity, from malnutrition, from diseases like nerve damage, or could be from bone fracture. So it can be many reasons. And the physical activity, there's two types of physical activity. One is anaerobic and the other one is aerobic. For anaerobic, you use fast fibers, and that's why you're using a lot of tension, but you cannot continuously do it. So that's why if you want to improve on that, you have to do it repeatedly. You have to have multiple sets and multiple reps, but you have to use higher weights, and that's way you can build muscle. And again, that depends on your goal. You can have higher strength, or you can have bigger size. That the type of activity varies. The amount of reps and sets that you do, that varies. But that's a different topic. And aerobic activities like walking, jogging, swimming, running, all those things, they are good for the blood vessels, for the heart, for the brain, for the lungs, and of course for the muscles. But these ones require slower fibers and continuous supply of the oxygen, so they need more myoglobin. Which one is better for weight loss? You have to do both. So muscle fatigue after you do some physical activity, the muscles are becoming tired. And why the muscle become tired? That can be because of lack of enough ATP. All the glycogens are gone, not enough oxygen is there. And there could be damage to plasma membrane or sarcoplasmic reticulum. It can be accumulation of hydrogen, so the pH is low, and also the muscle are tired, and then there's a pain there, the tendons are damaged, so the muscle become fatigued. So you need to rest. Another topic is called cardiac muscle, and they are called cardiocytes or cardiomyocytes. They should be aerobic because during anaerobic activity, the person will have a heart attack. So they need a lot of oxygen. They are not under your control, so they are involuntary. They have striation, as you can see. These are the striations, and they are branched. So the skeletal muscle, they were parallel to each other, but this is branched, and they contain intercalated discs. So this is intercalated disc that separates one muscle cell from another. They have a lot of mitochondria, as you can see. That's why this aerobic, so it needs a lot of mitochondria, and you can see here the mitochondria as well. Most of them has one nucleus. Some of them may have more than one nucleus. Compared with the skeletal muscle, they all have multinuclei. And the muscle fibers, it is surrounded by sarcoplasmic reticulum, but there is no cisternae here. So there is no triad. The T-tubules are wider than skeletal muscle. Okay, so there's some differences there. So the intercalated disc, these are the intercalated discs that you can see, and it's good way to recognize and differentiate this muscle from other type of muscles. The cells are branched. You can see the branching. They are not parallel to each other. Okay, most of them has one nucleus. They have one nucleus. Sometimes maybe more than one nucleus, and they are separated from each other by the intercalated disc. So this is located in the Z line, and it contains gap junction. The gap junction, if you remember it, it moves small ions and also lipids. In this case, sodium can move from one cell to another cell, so that improves efficiency, and not all muscles cells need to be innervated by a nerve. So some muscle cells are innervated, but the other ones are activated through the gap junction. There is also desmosomes are there that is for support and attachment of the muscle cells with each other. They have a lot of specific characteristics. One is automaticity, which means that they have their own pacemaker cells. They are similar to nerve cells that they can activate and that can change membrane potential, and that will stimulate muscle contraction. But they are also influenced by the nerves that are coming from the brain. These ones can increase or decrease cardiac activity, depends on the demand. For example, you're running, you need more oxygen, you need more muscle activity, so the heart rate increase. But at rest, you don't need that much, so your heart is relaxed and does not use that much contractility. The contraction time is longer than a skeletal muscle because you want all the blood or most of the blood to pump out of the heart, and also the refractory period is longer because the heart should be relaxed so it can get blood from the blood vessels. And if this is very short, then the heart will not fill up, and then it will affect the ejection or the amount of blood that is coming out of the heart, and it will affect the blood pressure. So heart is contracting 1/3 of time and relaxing 2/3 of the time. Also, the sarcolemma is a little bit different than skeletal muscle. Here, the sarcolemma is has more permeability to extracellular calcium, so more calcium can enter the cell. In the skeletal muscle, majority of the calcium was coming from sarcoplasmic reticulum, but here from both sarcoplasmic reticulum and also extracellular calcium. So this is why when the amount of the calcium changes, especially if it is low, then it will affect the cardiac activity, and that's not a good thing because you don't want heart rate and rhythm to change abnormally. This is very serious condition. Smooth muscles, they're located in different parts of the body. One is in the integumentary system, if you remember from chapter 5, the erector pili muscles that are attached to the hair, and that can cause goosebumps during fear. For example, I don't care much about it. If you really care a lot, you can go and watch the Goosebumps movie. I'm joking, guys, okay? The other one is about the blood vessels. So the blood vessels are covered by epithelial tissue, but also by smooth muscles. So that's important for the blood pressure. Also for the airways, the amount of oxygen that is needed. So if you're exercising, then the muscles relax, more oxygen enters the airways. If you're relaxing, then those muscles contract, so less air goes inside. It is in the reproductive system, it can carry a sperm or ovum, and in the glandular system, it can contract and eject secretions, exocrine secretions. In the digestive system, it carries or moves food and feces. In the urinary system, it can move urine. It also has sphincters. These sphincters are like gates. These are circular muscles that prevents movement of different material from one area to another area. For example, movement of urine from the urinary bladder to outside. So then that way, when you're consciously aware that the bladder is full, then the person can goes and urinates, and then that can relax those muscles. Those are preventing movement of urine or feces or food from one area to another area. So the smooth muscle is spindle shaped. It's not striated. It's not under your control, so involuntary. It has only one nucleus, and it has thick and thin filaments. but you can see that they are not close to each other, so they are in random locations, and the thin filaments are attached to these structures, proteins. They are called dense bodies, and myosins usually have a lot of heads because they are not very close to each other, so that's why they have more heads. During contraction it can attach. The smooth muscles, they don't have tendons or aponeurosis. It means they are not attached to other muscles, or they're not attached to the bones. So here that you can see that the dense bodies are still there, and they are connected with each other by intermediate filament that's called desmin. Okay, they are connected to each other. Okay, and also that's inside. So outside, these dense bodies are connected to each other too, and when there is muscle contraction, then they all contract. So these contraction from one cell, it goes to another cell, to neighboring cells, and the whole cell will contract. I mean, the whole tissue can contract at the same time. So if you remember it, as I said in the previous slide, in the dense body inside, you have the thin filaments. So that is when they're attached with the thick filaments, then it can cause contraction. So when the calcium enters, that calcium attaches to calmodulin instead of troponin that was in the skeletal muscle. This is calmodulin, and that will activate myosin light chain kinase. That will break down ATP, and the muscle will look like this. Okay, so if you have any question, please let me know. Thank you.